Okay, good morning, everybody. My name is Dave Van Sanford, wheat breeder at UK in the Department of Plant and Soil Sciences. And I'm really happy that you stopped by this morning and tuned in. I'm going to be talking about the wheat breeding program for a bit, and then Bill will be talking about the variety testing program. So I just wanted to give you a little sense of what the breeding program is like. And I'll start by echoing what both Chad and Carl Bradley said regarding support. We've had very long-standing support from the Kentucky Small Grain Growers Association, and without that support, there would be no breeding program. We've also been very fortunate to receive some support from Seymour Milling and also from the U.S. Wheat and Barley Scab Initiative. So when you think about a breeding program, it's often shown in this way of a triangle, very wide across the bottom, and this depicts kind of the timeline of a developing variety. Excuse me. So we start out and we make typically 400 to 500 crosses a year, and then from those crosses we have about 500 F1 families, and over time we go through a process of inbreeding and selection. And so by the time we get here to the yield trial stage, we often have 500 to really, in our case, it's about 1,500 lines that we're going to put into yield trials. And it's at this point that we might do some genomic selection. I'll talk about that in a little bit. Now we're looking at the usual suspects in terms of traits: grain yield, test weight, plant height on down the row, certainly head scab, as Carl talked about, and milling quality is also on our list of must-have traits. As we continue to inbreed, we get to a stage here where we have maybe 20 lines in our program that we put in these collaborative nurseries. And that's something we're real lucky to have in the eastern wheat region in the US, where breeders test each other's lines in these cooperative nurseries. We get agronomic data, disease data, and milling and baking quality data as well. And then I typically put 10 to 12 lines in Bill Bruening's variety trial. And those are the best of the best over several years of testing. Now, I want to just give you a sense of what is involved in plant breeding. How do we begin this process? So crossing is where we begin. And this is a close-up of a wheat flower, or floret as we call it. The yellow anthers are what shed the pollen onto this fluffy white stigma. Pollen grains land there, germinate, tube grows down, and then on all the way to the ovary, and that's where the seed development begins. Now, because we have perfect flowers in these self-pollinated crop, like wheat, that is, they have both male and female parts, we have to remove the anthers if we want to do crossbreeding. And so we do that with forceps, and then we pair those female heads up with male heads in these clear bags, as I've shown here. So this is in our greenhouse in Lexington. You can see anthers hanging from the intact male head, and the pollen has been spread onto these heads below that were turned into females, and we have here brand new F1 kernels that have developed and are still growing. So these seeds and seeds from other generations that we've selected are put into these plastic planting trays, and we plant those seeds, all the seeds from one head, into what we call a head row, which is about 4 feet long. And we grow about 20 or 25,000 of these at Lexington every year from all of the generations, from F1 on up to about F10. These have to be sickled and then threshed. And here you see a crew going through. Here's a young woman with a sickle, and we take heads from each row, or we bundle the entire row, load all the bundles on a wagon, and this process occurs many, many times. And then we take all the bundles to our greenhouse, which during the hot summer months is not occupied with growing plants, fortunately. So it's a good place for us to store these bundles and just let them be out of the weather while we get ourselves out of the field, which usually takes several weeks. After that point, then we'll begin to thrash all of these bundles. So just to give you a sense of the footprint of the program, this is a map of Kentucky, and this is where we're located. So our home farm, Spindletop Farm, is in Fayette County, and there we grow everything. All of our yield trials, all of our head rows, our double haploids, fungicide by variety test, our scab nursery, seed increases. Nearby Woodford County, we have many of our yield trials, including our collaborative tests that I told you about earlier. And then down here at Princeton, where I am today, we have, again, all of our yield trials and a number of our F2 populations and some specialty tests. And then Chaco, we're actually on a farmer's farm at Walnut Grove Farms in Logan County. And again, mostly yield trials, some of our F4 populations. And you note I've got a couple of arrows pointing to a seed increase. This is a line that I'll talk about a little later that we're hoping to release next fall. We've got one increase of about 25 acres down in Logan County, and then about 30 or so acres in Washington County. So you heard Carl Bradley talk about head scab earlier. It's a big, big part of our program because we focus very heavily on head scab resistance. And so this process begins for us during February when we begin to grow the fungus that causes head scab in Petri dishes. And once that is grown across the whole dimension of the Petri dish, we put corn in these metal tubs, let it imbibe water overnight, and then we cut up the Petri dish medium and just put it in the tubs. And after a short period of time, the fungus has colonized all of the corn, which is what we want to happen, so that it can serve as a vehicle for inoculating our scab nursery. So this is a still photo of our scab nursery in Lexington, and you can see that we have these very fine misting nozzles that create a mist that really envelops every head in the nursery. So what we're trying to do, and this will be after we've distributed the scabby corn between the rows, what we're trying to do is basically create a scab epidemic within the nursery so that we can really ratchet up the disease pressure and find the most resistant types possible. So we have all of our breeding material in here. We have Bill Bruening's variety trial. We have a bunch of graduate student studies where we're looking at the inheritance of head scab, all kinds of stuff. Now this is just a little video clip that'll kind of show you what the mist looks like when it's running. It's not a great view, I realize, but it kind of gives you a sense of just what a fine mist we end up with. So as Carl said earlier, head scab, Fusarium head blights caused by this fungus, Fusarium graminearum, occurs at flowering and afterwards, as he said, on up to the soft dough stage. Warmish temperatures are preferred and high humidity. Again, the symptoms, same kind of thing we saw from Carl there. These are the kernel symptoms, and these pinkish white kernels that we call tombstones are just loaded with this mycotoxin, many of us call vomitoxin. It's a real concern of ours from a food and a feed safety standpoint, but certainly from the dockage that the farmer encounters at the elevator. So it's the most serious aspect of head scab. Now, as the season wears on and the material in the nursery begins to senesce, it's kind of looking like this. And this is the point about 21 to 28 days after flowering, as grain fill is ending, when we read our symptoms. This is the kind of variety of responses we see. You can see this row, there's a lot of partially bleached or fully bleached heads in this row. You want to get it at a stage when you can still see some green material so that you don't confuse the bleaching with natural senescence. This row looks better in terms of more green material than white. And here's a row of our susceptible check and spreader variety, the old variety Pioneer 2555, which was a great early variety, high yielder, but it was very susceptible to head scab. And so this is what we're looking for. When we see 55 looking like this, we know that we've got a good infection and we're going to get some really good data out of the nursery. We evaluate the scabby seed by creating these visual references. So we basically we add sound kernels and scabby kernels together in these proportions from 5% up to 90% scabby kernels. And that gives us some standards that we can use to judge what comes out of the nursery. So this is a subset of the varieties that we have evaluated over 4 years' time, from 2016 through 2019, for 2 traits, FDK, which is fusarium damaged kernels, and then DON or vomit toxin. We see up at the top Truman, late variety out of Missouri, very good scab resistance. So we see it on the low end, about 14% fusarium damaged kernels and 6% or 6 parts per million DON. And down here at the bottom, the Pioneer variety, on up to 15 parts per million DON. And this is the line that we were increasing that I mentioned earlier, this 1145 line. So it tends to be lower in DON historically over the years that we've tested it, which is exactly what we're looking for. Now, I'm just going to shift gears for a minute and tell you about a couple of other things we're doing in the breeding program that are exciting and I think have a lot of potential for growers. Genomic selection is something that has really come into play in the last 5 years in plant breeding. And basically, we use a statistical model and DNA sequence data to predict how untested new lines are going to perform in the field. So the idea is this: we have recent yield trials of lines where we have field data and DNA data. We use that information, create a model, and these new lines, we have DNA sequence data, but no agronomic data yet. We use that model to estimate their variety potential, and based on that, we decide which ones we're going to take to the field and test, because yield trials are very expensive. So this is something that promises to really reduce cost and time for us. And this gives you a sense of the predictive value of these models. So for yields, we've gotten as high as 64% accuracy in terms of what was predicted to be good and what was actually good. Fusarium head blight, the DON, for example, 57%. This will be very important for us because we cannot fit everything that we test into that scab nursery. So we can eliminate the lines that are predicted to have very poor scab resistance based on the model. The other thing that we're looking at that's exciting to us is speed breeding. That's one of our real impediments. We work with winter wheat. It's got a vernalization requirement, and it takes a lot of time, and we usually can only get 2 generations a year. But if by using these special LED lights, we're able to get Truman. It's photoperiod sensitive, for one thing. It's got a long vernalization requirement, and it's just late. We can get it seed to seed in about 100 days. So that's really good news, and we can therefore go to 3 generations a year, and that'll just speed up the breeding process. So that's exciting stuff to us. All right, I mentioned this 1145 line. This is a line that we've tested for a couple of years now in Bill's variety trial. Just to give you a little sense of how it's doing, it's not been up at the top of the yield trial, but it's hanging in there and competing very effectively with these varieties that have done very well in the variety trial and are widely grown in Kentucky in both years, varieties like Armor Rage, for example, the Agramax varieties. So the Kentucky line has excellent test weight, it's short-statured, and it's mid-early. And we've also tested it in Wheat Tech's trial. It's done very well there, competing and being in the same statistical class as some of the very widest grown varieties and most popular varieties in Kentucky right now. And we've also tested it in the Virginia State Virginia Tech, rather, yield trial, and it's competed very well there, exceeding Hilliard, Armour Mayhem, some varieties that have done quite well in Kentucky. In terms of scab resistance, it is hanging right in there with Truman and Pembroke 14. Pembroke 14 is our most scab resistant variety. It has FHB1, a single gene that really gives us a lot of resistance. The 1145 does not have a FHB1, but it's got 3 genes that combine to give very good resistance. And as I mentioned before, very low DON. So we're excited about that. We increased it for the first time on a farmer's farm in Washington County in 2019, and we're on a different farm in Washington County this year, and also down in Logan County. In both cases, it was planted late, so I think that's going to bode well in terms of escaping freeze damage. It's looking really good at both locations. So fingers crossed, we'll have some seed available this fall. And with that, I'll turn it over to my partner in crime, Bill Bruening. Unless there are any questions, I'll be happy to answer them.
Okay, well, good morning. And what I'd like to do here is share some information that I normally wouldn't be able to share with you in a field situation. There we go. And I'd also like to thank the Kentucky Small Grain Growers Association for supporting this program. Without their support, we would not be able to do the many unique trials, which I will discuss shortly. We simply wouldn't be able to do that without their support. So we thank them very much. And variety selection is considered the simplest, most cost-effective way to maximize production profitability. And these programs, be it wheat, corn, soybean, or whatever, thoroughly evaluate varieties and new genetic lines to provide unbiased results on varietal performance. So if somebody suggests that you purchase a variety of seed that isn't tested in a state variety testing program, the question is, do you really know what you're buying? It might not be $1 million question, but it's certainly going to be a $1,000 question potentially. So in this year's test, we have 88 varieties tested over 7 different locations. We have a test in Fulton County this year. Up in the Ohio Valley region, we're in Webster County. Of course, we have our usual test at Princeton. A couple along the southern tier, we're in Christian County. And then over at the Simpson-Logan County line, in central Kentucky, we have a location at the Woodford County Farm, as well as in Lexington. And this bulletin is available in printed format at all extension offices in the middle of July. You can get this data online typically around the 1st of July in this format, but usually we have the Excel files on our website at the end of June. So go ahead and look at that as needed. When making variety selection decisions, we recommend that growers utilize both the overall state summary table, which combines the data from all the different locations for a given year into one very robust table. Of course, when looking at yield, we also encourage you looking at 2- and 3-year data to really see how these things perform across so many locations and many years. You really get an idea of which are the most robust varieties. But we also recommend that you use your variety information from your regional test. I would say find the varieties that perform well both within your region, but also factor in, find those same varieties, make sure they perform well across the overall state summary as well. But it's our primary table. You're going to get your test weight information, height, often lodging information is included for most years, and also estimates of maturity. So this is the main table to look at. But you know, most of our interest is in grain production, of course, but some specialized tests we do. One is the wheat forage variety test, and this is the largest wheat forage variety test in the country. Last year, in 2009, we looked at 3 different things. We looked at a harvest at the soft dough stage, which you see are forage specialist harvesting our plots here. This is typically what most cattle producers utilize wheat forage for. At this stage, we tend to get a high amount of tonnage of what would be considered an average quality hay forage. And there are dramatic differences among varieties. Last year we saw at 1.8 to 3.1 tons per acre, depending on the variety. So, you know, if forage is part of your system, certainly look at varieties. You can dramatically increase your yields just by variety selection. Last year we also took a sample at boot stage, and here at this stage are primarily for those growing dairy forage, where the protein content is similar to alfalfa, much higher energy level, but the lower yields. And with the boot stage, you can do full season corn, full season soybeans right after it. Soft dose stage, you can certainly do full season soybeans following that, and possibly full season corn there. And last year, we also started taking samples to determine the varietal cover crop potential, or would also be considered varietal differences in winter grazing potential, basically seeing which varieties produce a lot of biomass to give you that good ground cover protection. Obviously, 19 is almost twice as much as 10 here. So again, varietals, there are varietal differences in all sorts of these different parameters as it relates to forage production. This is also a good table to access information on whether the head type of a variety, whether it's bearded or awnless. Cattle tend to do better on awnless varieties because sometimes the awns can irritate their digestive tract. So we tend to recommend these, particularly at the soft dose stage. It's not going to matter when you're harvesting at the boot stage, but at the soft dose stage, if it gets a little fibrous, it can cause some trouble. Also, we found out last year we lost a test of deer damage, and we found that the deer went in at one Ohio Valley location last year and ate almost all the heads off of our awnless varieties. We lost a test, but we got some good information on deer damage. So I would say if you're in an area that's prone to deer damage or high deer populations, I would recommend that you utilize a variety that is not awnless. Go with bearded varieties. Another test is our wheat variety straw test. Again, there are dramatic differences in yield here. We see you can almost triple your tonnage per acre in terms of straw production. Here in central Kentucky with the equine industry, certainly growers are marketing both their straw as well as their grain. On the other side of the coin, growers in western Kentucky might be more eager to just have a low residue, low straw yielding variety that produces high grain yields. They get that double crop soybean established just as quick as possible. A new test we established last year, and we are continuing this for a second year currently at Princeton, is a metribuzin tolerance test. And metribuzin can be a very effective herbicide for ALS resistance weeds. However, some varieties are susceptible to metribuzin damage. And we did in fact see this last year when applying the maximum labeled rate. We were able to rate injuries on a scale from 1 to 5, where 5 we saw severe necrosis. This is the slide of the worst variety we had out there. And this is the differential line between the healthy part of the plot and the part that was sprayed with the herbicide. And but we also had varieties that went all the way down to a 1 that showed absolutely no susceptibility to metribuzin damage at all. And it did affect yields. Again, just half the plot was sprayed, and we still saw a 40% reduction here with this one that was hammered. No differences in test weight. And the more severely damaged lines, we saw that the height tended to be stunted compared to the control, and we also saw that the maturity seemed to be delayed, at least in terms of heading date. So this is something that seed companies requested that we look at. There's been a lot of interest in this, and we were able to start making it happen to hopefully get this information out to growers. But like Dave's been talking a lot about diseases, Carl Bradley spoke a lot about diseases in wheat, and we do rate our varieties at specific non-fungicide locations for disease reaction. We can only rate those pathogens which are prevalent in a particular year. So Carl mentioned we might be seeing some stripe rust coming in. We'll see whether we can get some ratings there or not this year, but we can only rate what's prevalent. If you're looking for variety information on disease reaction, if you don't see it in this year, for example, in 2019, get online and go back through some of our more recent ones. Look at 2016, 17, 18, see if those varieties are there. We might have different ratings for gloom blotch, stripe rust, leaf rust, a lot of other information available. So just another source of information to help you either with your crop management or in terms of variety selection. We have been conducting barley research for many years at several locations in collaboration, mostly with Virginia Tech University, but we're starting to work with some private companies as well too. We test 3 different types of barley. Of course, traditional barley has its hull primarily used for animal feed. It works very well in helping move their feed through their digestive tract, having that fibrous hull. But we've also been working with Virginia Tech with hulless barley, which sheds the hull during the combining process, and you end up with an energy-rich grain that looks very similar to wheat. So there aren't very many varieties available, but we do continue to work on this, try to find new uses for these crops, and hulless barley may be one of those crops for the future. But one thing that's been generating more interest as of late is malting barley varieties, and the double asterisks indicate the lines which are considered malting barley varieties. And you can see there's a range in yields. But one thing I would point out is that we're seeing is a lot of these malting lines tend to mature later than what we're used to barley maturing. You know, we get a great double crop soybean yield bump by being able to plant barley and harvest it earlier. But what we're seeing with some of these malting lines is that they're maturing a little bit closer to wheat. So you need to factor that into your decision, certainly in terms of variety selection, but also in terms of your overall crop production management scheme. Okay, Triticale is the result of a wheat by rye cross. And this was created by breeders about 100 years ago. And its goal was to create a crop with the baking qualities of wheat, but rye's disease resistance and tolerance to environmental stresses. And Triticale is commonly used as a forage crop. You can harvest it up to the boot stage and get great quality forage. Thereafter the boot stage, though, once it starts to get into that reproductive period, it gets very fibrous very quickly and loses its quality very quickly. So that's something you need to watch with forage production for Triticale. It's an excellent cover crop. I find it grows even more vigorously than rye itself. Very big, broad foliage. But in more recent years, we're starting to see some interest in brewing, microbrewing, as well as distillation uses for Triticale. So that's why we're taking a closer look at Triticale variety testing. It's taller than wheat, typically around 4, sometimes 5 feet tall. It stands a lot better than rye. And its heading date, it matures around the time of a full season wheat, or possibly, I guess, a medium to medium light maturing wheat. And the next test we'll talk about is cereal rye, branching off of our Triticale talk. So with the rye, of course, is commonly grown as a cover crop in Kentucky. And what we're looking at here with our varieties, we're evaluating hybrids and our traditional rye that we've been growing for many, many years. And we're seeing big differences in the hybrids versus the traditional rye. You look at the yields here, all between 60 and 70 for the hybrids, and you look at the dramatic drop off up to the traditional rye. So big advantage in terms of yield for these hybrids. The hybrids tend to be shorter. They're typically 4 to 5 feet tall compared to your older rye or non-hybrid rye, which are 5 to 6 feet tall. Big differences in yield potential. We didn't see a big problem with lodging last year, but typically these numbers, instead of ranging from 10 to 20, typically range from 20 to 70% lodging for rye. It's a terrible problem as part of the production process for growers, and hybrids have really done a good job of having good stem strength. One thing I will point out about rye, yeah.
Now you've got a question from Terry Mashburn. He's asking about heading date.
Mm-hmm.
You could explain that heading date number.
So the heading date number, for whether it's wheat or rye or everything, I take heading notes at all locations, all of our plots, all of our varieties for every crop, and estimate the heading date. And here it's listed as days after April 1st. So in this case, this would actually be May 5th, May 6th, May 8th. And that's the day that 50% of the heads have emerged from the crop. So that's what we use as to define heading date. And that is just an estimate of the plant's ultimate maturity. And it transitions, Terry's question brings us into the point that these hybrid lines do mature later than your traditional rye lines. You're going to get a double crop soybean yield advantage if you can harvest earlier rye, traditional rye, versus going with hybrids. So when you start working with hybrids, you need to factor this into your decision as well, because it is going to bump you back to basically the similar time that you're harvesting your wheat crop too.